Welcome to Deep Dive. Today, we are taking an exhaustive journey into a world that is, well, fundamental to life itself,
Yeah.
the histology of blood.
It's a topic that I think for most people is just, it's the red liquid in our veins.
Right. But as the comprehensive chapter we've pulled from Histology, a text in Atlas makes so clear, blood isn't just a fluid, it's a highly specialized fluid connective tissue.
A tissue, exactly. And it's circulating continuously through this closed network of the cardiovascular system.
The scale is pretty amazing when you stop to think about
Oh,
it.
it really is. For an average adult, you're looking at around 6 liters of this tissue. That makes up something like 7 to 8% of your total body weight.
So our mission today is to go way beyond that red liquid idea.
Yeah, our goal is to systematically walk you through the entire complexity of this tissue, from its basic components and functions all the way to the intricate factory that produces it, the bone marrow.
I think defining those functions up front is important because there are so many of them. It's not just about oxygen delivery.
Not at all. It's a whole ecosystem. Beyond transporting O2 and CO2, blood is managing nutrient delivery, waste removal. It's a transport system for hormones.
And it maintains stability, right? Homeostasis.
Critically, it maintains what we call homeostasis by buffering pH, regulating temperature, and of course, executing the immune and coagulation responses.
And all of that complexity is packed into just two major parts.
That's right. You have the formed elements, so the cells and cell derivatives, and then you have the protein-rich liquid, the extracellular matrix, which we call plasma.
To really get a handle on the basic composition, we can look at what happens when you physically separate those two parts.
Exactly. And that brings us immediately to a key clinical measurement, the hematocrit,
Or HCT.
the hematocrit, or HCT, sometimes called the packed cell volume, or PCV.
Yeah.
It's really the foundational diagnostic tool here.
So you just spin the blood down.
You literally centrifuge an anticoagulated sample, and it gives you the percentage of the total blood volume that's occupied by the packed erythrocytes. That physical separation just instantly shows you the volume fractions.
And when you look at those volumes, the red blood cells are a huge part of the cells, but the plasma is still the majority of the total volume.
Generally, yeah, you're looking at a ratio of about 45% formed elements to 55% plasma.
But it varies a bit.
It does. The specific numbers differ a bit between the sexes. HCT ranges are typically 39 to 50% for men and 35 to 45% for women.
And if you look at figure 10.1 in the text, which shows this centrifuge sample, it's so clear that bottom layer is almost entirely red blood cells.
Almost 99% of that packed bottom layer is erythrocytes.
But there's that tiny little middle layer, which is, I think, the visual confirmation that the white blood cells and platelets are just. They're numerically insignificant compared to the red cells.
That's the Buffy coat. It's that thin, light-colored layer you can see between the plasma and the packed red cells. And you're right, it's tiny. It only accounts for about 1% of the total blood volume.
But everything else is in there.
Every single circulating leukocyte and platelet is in that 1% sliver. And clinically, knowing that HCT value is vital, if that packed cell volume is low, it's a very strong indicator of anemia.
And when we talk about how dominant the erythrocytes are numerically, the raw data from table 10.1 really drives the point home.
It's genuinely astonishing. We are talking about 5 times 10 to the 12 erythrocytes per liter of blood.
5 trillion.
5 trillion per liter. Now, contrast that with the leukocytes, which are hovering around 7 times 10 to the 9 per liter.
So 1,000 times more.
Almost 1,000 to 1 ratio. The oxygen carriers outnumber the immune cells by a factor of nearly 1,000. Functionally, white blood cells are critical, but numerically, they're just patrol vehicles in a sea of freight ships.
Okay, so let's unpack the liquid environment those freight ships are navigating. The plasma. We know it's about 55% of the volume. Table 10.2 breaks it down chemically.
Chemically, plasma is the extracellular matrix of blood, and it's mostly water, about 91 to 92% water.
And the rest?
The remainder is about 7 to 8% proteins, then a tiny 1 to 2% of other essential solutes, electrolytes, nutrients, hormones, dissolved gases. A fundamental role of this fluid is just maintaining the optimal pH and osmolarity for the cells.
But it's really the proteins that define plasma's function. We have to talk about the big 3 plasma proteins.
Absolutely. And almost all of these are synthesized and secreted by the liver. We can start with albumin. It's the most abundant and also the smallest, about 70 kilobar.
And this is the protein that I've heard described as the water magnet in our vessels.
That is the perfect way to put it. Albumin's main job is generating what we call colloid osmotic pressure on the vessel walls.
Okay, and what does that pressure do?
That pressure is the mechanism that ensures fluid, is drawn back into the capillaries from the surrounding tissues. It's what maintains the critical fluid balance between the blood and the tissue's extracellular fluid.
So if a patient has a condition that causes them to lose albumin, like, say, severe liver disease or kidney failure, the consequence is something you can actually see.
Precisely. If that osmotic pressure drops because there's less albumin, the hydrostatic pressure in the vessel forces fluid out, but it fails to come back in.
So it just accumulates in the tissues.
Yes. It causes fluid accumulation edema, which you often first notice as swelling in gravity-dependent areas, like the ankles at the end of the day.
And albumin has other jobs, too.
Oh, yeah. Beyond its osmotic role, it's a major carrier molecule for things that don't dissolve well in water, like steroid hormones, bilirubin, and even a lot of pharmaceutical drugs.
Okay, next up we have the globulins, which is a much broader, more diverse family of proteins.
We can categorize them into two main groups. First, you have the immunoglobulins, which are also known as gamma globulins.
Antibodies.
These are the antibodies, and they're not made by the liver. They're synthesized by specialized immune cells, called plasma cells, and they're critical for the humoral immune response.
And the second group.
The second group are the non-immune globulins, the alpha and beta globulins. These are secreted by the liver, and they mostly serve as carrier proteins, things like transferrin for iron, ceruloplasmin for copper, and specific lipoproteins. They also include some key components for blood clotting.
And the largest of them all, fibrinogen, is the ultimate specialist for that clotting process.
Fibrinogen is huge, 340 kilodalton, and it's also liver-derived. Its entire purpose is hemostasis.
So stopping bleeding.
Right. When a vessel is breached, a rapid cascade of reactions is initiated, and that cascade transforms soluble fibrinogen into its insoluble active form, fibrin.
And that's what forms a clot.
These fibrin monomers then polymerize incredibly fast, forming a cross-linked dense net that acts as an impermeable barrier to stop blood loss.
Since fibrinogen is always present, if we just leave a blood sample to sit, it's going to clot. And that leads to this really essential lab distinction between plasma and serum.
That's a key point of clarity. Serum is essentially plasma after the clotting factors, including fibrinogen, have been used up during coagulation.
So if you want to study the whole thing, you have to stop it from clotting.
Exactly. If you want to analyze the entire protein content, you must add an anticoagulant like citrate or heparin right when you collect the sample. That prevents the cascade, leaving you with plasma. If you let it clot and then separate the fluid, you're left with serum.
And just to quickly connect the blood to the rest of the body, the interstitial fluid is basically modified plasma.
Yes. The fluid bathing our tissue cells is derived from plasma filtered across capillary walls. The basic electrolyte profile is similar, but its precise composition is heavily modified by the specialized barriers it encounters.
Like the blood-brain barrier.
For instance, yeah. The tight controls of the blood-brain barrier drastically alter the chemical makeup of the interstitial fluid in the central nervous system.
Now, to study all these cells in detail, we can't use the standard H&E staining that we use for fixed tissues. We rely on a specialized technique, the blood smear.
The blood smear is unique because we skip all the fixation and embedding. You take a tiny drop of blood, you spread it across a slide to create what we call monolayer. Figure 10.2 in the text illustrates this, and then you air dry it immediately.
The goal is to see every cell individually.
Right. To see them clearly and avoid any cell overlap.
And the stain is what gives us that distinct blue, purple, and pinkish red contrast that we all associate with looking at blood under a microscope.
That contrast comes from the modified Romanowski-type stains. These use a combination of basic dyes like methylene blue and its oxidized derivatives, the azures, and an acidic dye, eosin.
So different parts of the cells pick up different colors.
Exactly. The basic dye stain structures like nuclei, RNA, and basophil granules a blue or purple color. The acidic eosin stains hemoglobin, and the specific granules of eosinophils a distinct pinkish red. This combination lets you immediately classify leukocytes based on their nuclear shape and granule color, which you can see perfectly in plate 10.1.
All right, let's move to the most prominent formed element, the erythrocytes, the red blood cells, defined by their specialization. They're enucleate, biconcave discs, about 7.8 micrometers across.
That structure is the absolute epitome of form-following function. Lacking a nucleus in most organelles means they are devoted solely to gas transport.
And the biconcave disc shape is key.
It's crucial. It dramatically increases the cell's surface area up to 140 square micrometers relative to its volume. This just maximizes the efficiency of O2 and CO2 exchange across the membrane.
And for any histologist looking at a tissue section, the consistent size of the red blood cell makes it an essential reference point.
It's the intrinsic histologic ruler. Since its size is so uniform, always about 7 to 8 micrometers in fixed sections, you can use it to estimate the size of anything nearby, whether it's other cells, collagen bundles, or even muscle fibers.
And they have a surprisingly short lifespan, only 120 days, so they have to be constantly replaced.
That's right. About 1% of the entire population is removed from circulation every single day. Most of that happens through what's called extravascular hemolysis, where they are phagocytosed by macrophages, mainly in the spleen, liver, and bone marrow.
Only about 10% just break down in circulation.
Yeah, the vast majority are actively removed.
We often see these images, like in figure 10.4a, showing them squeezed almost unbelievably through narrow capillaries. They must be incredibly flexible.
That flexibility or deformability is absolutely non-negotiable. They have to be able to squeeze through capillaries that are often narrower than 3 micrometers.
So they have to fold up.
They have to fold, deform, and then immediately snap back into their biconcave shape as soon as they exit that stricture. Figure 10.4b shows another common pattern you see, rouleau formation, where they stack up like coins.
You see that mostly in a lab setting, though.
Mostly in vitro, yes, but you can sometimes see it in vivo if plasma protein levels are unusually high.
And given that they lack internal organelles, they have to rely entirely on a specialized protein scaffolding right under the membrane to get that strength and flexibility.
It's an incredible piece of microscopic engineering, and it's detailed in figure 10.5. The whole mechanism relies on coupling the cell membrane to a flexible, self-assembling internal framework. We could divide the components into 2 groups.
Let's start with the anchors,
Yeah.
the integral membrane proteins.
The 2 major integral families are the glycophorins and the band 3 proteins. Glycophorins, especially glyphorin C, provide anchoring points for the cytoskeleton.
And the band 3 protein is even more important.
Arguably the most critical and abundant. It's a transmembrane protein that serves as the major site for attaching the internal cytoskeleton, and it's also where hemoglobin binds transiently.
And we should note that the parts of these proteins sticking out of the cell carry the entire identity system.
Indeed. The specific carbohydrate structures attached to those extracellular domains of the glycophorins and band 3 proteins, those are the blood group antigens. We'll get back to those in a moment.
Okay, now for the internal frame, the peripheral membrane proteins.
This internal framework is organized into a 2-dimensional hexagonal mesh. The key structural unit is spectrin.
And these chains link together?
Yes, the alpha and beta chains of spectrin form long, flexible rods that link together. The spectrin lattice is then secured to the lipid bilayer via 2 major complexes. The band 4.1 protein complex, which links spectrin to glycophorin C, and the anchorin protein complex, which links spectrin to that massive band 3 protein.
So it sounds like a really sophisticated suspension system. If any one of those connection points has a genetic flaw, the whole thing just collapses.
It collapses dramatically, and that leads directly to hereditary erythrocyte disorders. Take hereditary spherocytosis, for example.
What happens there?
That results from a defect in the anchoring proteins, typically in that anchorin complex. When the anchor fails, the membrane gets destabilized, and pieces of it just peel off.
So the cell shrinks.
It loses membrane surface area while keeping its volume, which forces it into a fragile spherical shape. And these spherical cells are rigid and get rapidly destroyed by the spleen, a process we call hemolysis.
And a defect in the elastic rods themselves leads to that elliptical shape.
That's hereditary elliptocytosis. This is usually caused by mutations in the spectrin protein itself. The mutated spectrin fails to let the cell rebound properly after being deformed. Over time, the constant stress causes the cell to progressively elongate into an ellipse. In both cases, the inability to withstand shear stress is what leads to the cell's premature destruction.
That discussion of integral membrane proteins beautifully sets up clinical correlation 10.1, the ABO and Rh blood group systems.
These systems are, of course, critical for blood transfusions in pregnancy. The antigens A, B, and O are actually short chains of glycoproteins or glycolipids attached to those integral proteins we just talked about.
And figure F10.1.1 shows that the difference between A and B type comes down to just a single enzyme.
Exactly. Which enzyme you express determines which carbohydrate gets added to the core O antigen. The A allele encodes an enzyme that adds N-acetylgalactosamine, and the B allele's enzyme adds galactose.
And if you're type AB, you have both. Type O, you have neither.
If you're type AB, you express both enzymes. If you're type O, you lack functional transferase enzymes entirely, so only the core O antigen is exposed.
And your immune system creates antibodies against the antigens that you don't have.
Yes, and that is the definition of compatibility. Type A blood has anti-D antibodies floating around in the plasma. This makes the type AB individual the universal acceptor because they don't have any circulating anti-A or anti-B antibodies.
And type O is the universal donor.
Right, because their cells lack A and B antigens entirely. Transfusing incompatible blood leads to catastrophic, immediate hemolysis.
The Rh system is simpler, based on just one key antigen, the D antigen.
Simpler, but equally critical. Rh positive means the D antigen is present. Rh negative means it's absent. The main clinical concern here is Rh incompatibility in pregnancy.
Right. An Rh negative mother carrying an Rh positive baby.
An Rh negative mother might be exposed to the fetus's Rh positive cells during delivery of her first Rh positive child. This can cause her to become sensitized and produce anti-D antibodies.
And that's harmless during the first pregnancy, but it creates a problem for any subsequent pregnancies.
Exactly. Those anti-D antibodies, which are a type of immunoglobulin called IgG, can cross the placenta in later pregnancies. They attack the fetal red blood cells, causing severe hemolytic anemia known as erythroblastosis fetalis.
But there's a preventative measure.
Yes. The mother is given RhoGAM, which is a dose of anti-D immunoglobulin around the time of delivery. This immediately destroys any circulating fetal Rh-positive cells before the mother's immune system can generate its own memory response.
Let's pivot to what's inside the erythrocyte. Hemoglobin, or HGB. This is the protein that defines the cell's function.
HGB is a 68 kilino protein, and its job is complex. It needs to have a high affinity for oxygen in the high oxygen environment of the lungs.
And then it needs to let go of it in the tissues.
It has to radically lower that affinity to efficiently offload oxygen in the low oxygen environment of the peripheral tissues.
Its structure is essentially 4 major parts.
Figure 10.6 details the structure. It's composed of 4 polypeptide chains called globins, 2 alpha and 2 non-alpha chains, and each globin chain is bound to one iron-containing heme group.
And the iron is what actually binds the oxygen.
The iron atom at the center of each heme group is what reversibly binds one molecule of oxygen.
And those globin chains change significantly throughout development, which is shown in figure 10.7.
In the developing fetus, the dominant form is fetal HDB, HbF, which is made of 2 alpha and 2 gamma chains.
And it has a higher affinity for oxygen.
Much higher affinity, which is essential for drawing oxygen across the placenta from the mother's blood. After birth, gamma chain synthesis rapidly declines, and beta chain synthesis takes over.
Which leads to the adult forms of hemoglobin.
The primary adult form is HbA, which is 2 alpha and 2 beta chains, making up about 96% of the total. A minor fraction, about 1.5 to 3%, is HbA2, which is 2 alpha and 2 delta chains. In a healthy adult, HbF drops to less than 1%.
And defects in producing these chains lead to serious diseases like the thalassemias.
Yes, they do, which sets us up for some crucial clinical connections. Clinical correlation 10.2 looks at the indispensable diagnostic tool, HbA1c, used to monitor diabetes.
Right, so this is hemoglobin that has glucose stuck to it.
It's hemoglobin A that has become irreversibly bound to glucose. This binding is a consequence of high blood sugar. But what makes this test so invaluable is the 120-day lifespan of the erythrocyte.
Because the hemoglobin stays bound to glucose until the cell is destroyed, it provides a kind of time average.
Exactly. Since the red cell lasts about 3 to 4 months, the HbA1c level gives you a weighted average of the patient's blood glucose control over the preceding 2 to 3 months. It's much better than a single glucose test, which can fluctuate wildly.
And finally, clinical correlation 10.3 dives into anemia and specifically sickle cell disease. Anemia is just defined as low hemoglobin concentration.
Low HDB concentration, below 13.5 grams per deciliter for men and 12 for women. It just means the oxygen carrying capacity is impaired, often because of a reduced number of erythrocytes.
Which can be from blood loss or increased destruction or just not making enough.
Insufficient production is very common, yes. It often comes down to dietary gaps or absorption problems.
When not enough iron.
Deficiencies in iron, vitamin B12, or folic acid are common causes. A fascinating specific example is pernicious anemia.
What's that?
It's an autoimmune condition where the parietal cells in the stomach are destroyed. Since those cells produce something called intrinsic factor, which is necessary for vitamin B12 absorption, a B12 deficiency results, and that impairs erythrocyte maturation.
Moving to sickle cell disease, HPS, it really highlights just how delicate those globin chains are.
It is the prototypical single-point mutation disaster. A single base change in the beta-globin gene results in the substitution of the amino acid glutamic acid, with the amino acid valine at position 6.
And that tiny change causes the hemoglobin molecules to interact pathologically, but only under low oxygen conditions.
Exactly. When oxygen tension is low, the HbS molecules aggregate and polymerize into long, stiff, insoluble fibers. These fibers then distort the erythrocyte, forcing it into that characteristic crescent or sickle shape, which is clearly illustrated in figure F10.3.1.
And these sickled cells are rigid and fragile.
They're rigid, highly viscous, and mechanically fragile. The functional consequence is devastating.
So they get destroyed faster and they cause blockages.
Their lifespan plummets from 120 days to about 20, leading to chronic hemolysis. Worse, their rigidity and viscosity cause them to block small capillaries, leading to tissue infarction, acute pain crises, and even stroke.
That gives us a really complete picture of the freight carrier. Now let's shift to the immune system's cellular patrol vehicles, the leukocytes, or the white blood cells. They represent only 1% of the formed elements, but they manage all of our defense.
And we classify them into two main groups based on the presence of prominent lineage-specific granules. You have the granulocytes, neutrophils, the eosinophils, and basophils, and the agranulocytes, which are the lymphocytes and monocytes.
But it's important to remember that all leukocytes have some granules.
Yes, all of them have non-specific eosinophilic granules, which are essentially their lysosomes, full of hydrolytic enzymes.
We have to start with the most numerous, the neutrophils. Table 10.1 shows they make up about half to two-thirds of all circulating leukocytes.
They're easily identified by their size, 10 to 12 micrometers, and their hallmark feature, the multi-lobed nucleus, usually 2 to 4 lobes, which gives them their other name, polymorphonuclear leukocytes, or polymorphs.
And in female cells, you can sometimes see the bar body.
Right. The inactivated X chromosome, which appears as a little drumstick appendage on one of the lobes.
Their cytoplasm is a genuine internal chemical weapons laboratory, packed with 3 distinct types of granules.
It really is. And the contents of those granules dictate their killing efficiency. First, you have the azurophilic primary granules. These are the largest, darkest, and least numerous. They are true lysosomes.
And what's in them?
They contain crucial components like myeloperoxidase, MPO, which we'll see soon is essential for producing bleach, powerful defensins, and cathelicidin.
Then you have the workhorse granules, the specific secondary granule.
These are smaller, much more numerous, and they often stain a light pink. They contain a cocktail of enzymes essential for degrading connective tissue and bacteria things like type V collagenase, gelatinase, and phospholipase, along with complement activators and lysozymes, and they're released first upon activation.
And finally, the tertiary granules help them move around.
The tertiary granules contain matrix metalloproteinases, or MMPs, like gelatinases and collagenases. These MMPs are critical because they help the neutrophil locally break down the extracellular matrix, which facilitates its rapid movement out of the blood and toward the site of infection.
That movement, diapedesis, is the single most important action for a neutrophil. They have to leave the blood to do their job.
They must. Diapedesis is the complex process of moving from the circulation into the surrounding tissue. It primarily happens in the postcapillary venules where blood flow is slower. Figure 10.9 lays out this two-phase molecular dance.
Phase 1 is the slowing down, the rolling stage.
The neutrophil expresses specific carbohydrate molecules on its surface, notably sialolysis. When there's inflammation, the endothelial cells start expressing proteins called selectins.
And they stick to each other.
The sialolysis binds weakly and transiently to the selectins, which causes the neutrophil to slow down and just roll along the vessel wall.
Phase 2 is the tight adhesion that leads to the full stop.
Right. Chemokines released at the inflammation site, like interleukin-8, activate specific receptors on that rolling neutrophil. This activation changes the conformation of neutrophil integrins, making them high-affinity binders.
And those integrins then lock onto the endothelial cell.
They bind strongly to corresponding immunoglobulin superfamily molecules, like ICAM1, on the endothelial cell, locking the neutrophil in place.
And once it's locked on, the neutrophil has to physically squeeze through that barrier.
This is an energy-dependent process. It can use the paracellular pathway, squeezing between the cells, or the transcellular pathway, piercing directly through the endothelial cell itself.
Okay, so once it's in the tissue, it has to recognize the pathogen, sometimes requiring this process called opsonization or coating before it can eat it.
Opsonization is basically tagging the pathogen for destruction. Neutrophils have an array of surface receptors, shown in figure 10.10, to recognize targets. They have FC receptors to recognize antibody-coded pathogens and complement receptors for complement-coded ones.
And toll-like receptors.
Yes. Toll-like receptors, or PRRs, recognize broad molecular signatures that are common to pathogens, known as PMPs.
Phagocytosis is then followed by this explosive killing mechanism, the respiratory burst, which uses highly toxic reactive oxygen intermediates.
This is the neutrophil's core bactericidal strategy, detailed in Figure 10.01. The engulfment of the pathogen triggers a rapid increase in oxygen and glucose consumption. This burst relies on two interconnected systems.
System 1 is the FOX system, or NADPH oxidase.
This is the essential first step. The NADPH oxidase complex gets activated and uses NADPH to reduce molecular oxygen into highly reactive superoxide anions. These anions are then converted into hydrogen peroxide and hydroxyl radicals.
And System 2, the MPO system, turns that hydrogen peroxide into something far more potent.
This is the key amplification step. Myeloperoxidase, or MPO, from those azurophilic granules catalyzes a reaction between the hydrogen peroxide and chloride ions inside the phagolysosome.
And the end product is bleach.
The end product is hypochlorous acid, which eventually degrades into highly toxic hypochlorite, which is literally chlorine bleach.
That's a fascinating chemical weapon.
Yeah.
And if that initial FOX system fails, the whole defense strategy just falls apart. That's the basis of clinical correlation 10.4, chronic granulomatous disease, or CGD.
CGD is an inherited immunodeficiency defined by the inability to generate that crucial respiratory burst. It's caused by a genetic mutation or absence of a component in that NADPH oxidase complex, the FOX system.
So they can't make the bleach.
They can't produce the necessary reactive oxygen intermediates, making them unable to efficiently kill the bacteria they've engulfed.
So the bacteria are engulfed but not killed, leading to these chronic recurrent infections.
Exactly. Patients are highly susceptible to recurrent bacterial and fungal infections. The body tries to wall off these persistent infections, leading to the formation of tumor-like masses called granulomas, a signature feature of the disease.
After the battle, the neutrophils die, and that accumulation of dead neutrophils and bacteria is what we see as pus.
And that striking yellow-green color we associate with pus, that's the visual evidence of the MPO enzyme's heme pigment.
There's also this other, more dramatic death mechanism called netosis.
It's incredible. The neutrophil can commit a kind of suicidal netosis, where it decondenses its own chromatin and ejects it, forming an extracellular web, or net, that physically traps and kills microorganisms, preventing them from spreading. It's a total sacrifice play.
Let's look at the next granulocyte, the eosinophil. They're less numerous, but highly specialized for specific threats.
Eosinophils are similar in size, but they're unmistakable because of their typically bilobed nucleus, shown in Figure 10.12, and their large, highly eosinophilic or pink-staining specific granules. The intense acidophilia really makes them stand out.
What makes the specific granules so toxic?
They contain a dense structure called the crystalloid body, and it's mostly composed of major basic protein, or MBP. This protein is what gives the cell its intense pink color, and it's cytotoxic, particularly to larger organisms.
So their target is usually something too large to phagocytose, like a parasite?
Yes. Their main function is cytotoxicity against protozoans and helminthoparasites. They also play a critical role in moderating allergic reactions. They release enzymes like histaminase and aryl sulfatase, which neutralize the histamine and leukotrienes released by mast cells, helping to dampen the inflammatory response.
Which is why a high count, eosinophilia, is a classic marker for allergies or a parasitic infection.
Precisely.
Finally, the least numerous of all, less than 0.5%, are the basophils, and they're functionally related to mast cells.
Basophils are often defined by their very coarse, dark basophilic granules, seen in Figure 10.13, which often completely obscure the nucleus. A unique feature is the presence of high-affinity FEC receptors on their plasma membrane for IgE antibodies.
And their granules are packed with the core ingredients for a hypersensitivity reaction.
They are the source of acute inflammation and allergic responses. Their specific granules contain heparin, an anticoagulant, histamine and heparin sulfate, which are potent vasoactive agents causing vasodilation, and leukotrienes, which are responsible for prolonged constriction of smooth muscle, especially in the bronchi.
So when an antigen cross-links the IgE that's bound to the basophil or a mast cell, the result is degranulation.
That rapid degranulation releases these massive amounts of vasoactive agents, which are responsible for the immediate and dangerous vascular changes you see in severe allergic responses, including anaphylaxis.
Now we shift to the agranulocytes, starting with the lymphocytes, the key operators of our adaptive immune system.
Lymphocytes are fundamentally different from the other white blood cells. They are primarily recirculating, and they are not terminally differentiated.
Meaning they can divide again.
They can re-enter the cell cycle, divide, and differentiate into effector or memory cells when they're stimulated by an antigen.
Morphologically, though, they look strikingly simple in a blood smear.
Figure 10.14 shows the classic appearance. Small cells, 6 to 15 micrometers, with a dense, intensely staining spherical nucleus that fills almost the entire cell. You just see this thin, pale blue rim of cytoplasm.
But functionally, they are a complex triad. We have T cells, B cells, and NK cells.
And you can't tell them apart in a routine smear. You need functional analysis and specific surface markers. T lymphocytes, or T cells, mature in the thymus and are responsible for cell-mediated immunity. They have T cell receptors and are subclassified into CD4 plus helper T cells and CD8 plus cytotoxic T cells.
And then the B cells handle the liquid defense system.
B lymphocytes, or B cells, mature in the bone marrow and drive humoral immunity by producing antibodies. The third type, natural killer NK cells, are part of the innate system, programmed to kill viral or tumor cells. They're typically larger and are sometimes called large granular lymphocytes.
And the complexity of the T cell subsets is staggering.
It's a vast system. You have the cytotoxic CD8 plus T lymphocytes that actively seek and kill infected cells. You have the helper CD4 plus T cells, which are the conductors of the immune orchestra, releasing interleukins to activate everyone else. And then you have regulatory T cells that turn the immune response off.
Next, the monocytes, the largest leukocyte, and the precursor to the massive tissue macrophages.
Monocytes average about 18 micrometers. Their nucleus is often large and characteristically indented or kidney bean shaped, as you can see in figure 10.15. They circulate briefly, only about 3 days.
And after circulating, they emigrate into tissue to transform into specialized phagocytes, creating the mononuclear phagocyte system.
Exactly. They transform into macrophages in connective tissue, Kupffer cells in the liver, alveolar macrophages in the lung, osteoclasts in bone, and so on.
And their function is twofold.
Right. They are highly effective phagocytes, and they are vital antigen-presenting cells, or APCs. They display degraded foreign antigens on MHC22 molecules to activate those helper T cells.
Let's connect the life cycle of the erythrocyte back to pathology before moving to platelets. Clinical correlation 10.5 focuses on hemoglobin breakdown and jaundice.
This is a direct consequence of recycling old red blood cells. When their 120-day lifespan ends, the hemoglobin is degraded. The globin chains are recycled, the iron is stored. The remaining hemoidy is chemically degraded into bilirubin.
And bilirubin has to be processed by the liver.
Exactly. The liver has to chemically modify or conjugate the bilirubin to make it soluble so it can be excreted into the bile. If the liver fails due to disease or a bile duct blockage, or if there's massive rapid hemolysis, like from a bad blood transfusion,
The bilirubin floods the blood.
right, and that results in jaundice, the yellow discoloration of the skin and eyes.
And for infants, the consequences can be much more severe.
In newborns, high levels of unconjugated bilirubin are toxic to the central nervous system. It can cross the underdeveloped blood-brain barrier and lead to a serious brain damage condition called kernicterus.
Now we address the final formed elements, the thrombocytes, or platelets. These are not true cells, but cytoplasmic fragments dedicated entirely to preventing blood loss.
They originate in the bone marrow from the largest cells in the body, the megakaryocytes. These cells are extraordinary. They become polyploid, reaching up to 64 amploidy through a process called endomitosis.
So they duplicate their chromosomes without dividing.
Over and over, making them absolutely massive. You can see one in figure 10.76.
The actual platelet release mechanism is incredible. It's regulated by the hormone thrombopoietin, or TPO.
TPO drives the maturation of the megakaryocyte. The mature cell then migrates right up against the bone marrow sinusoids and extends these long branch processes called proplatelets directly into the vessel lumen. Platelets are then shed from the ends of these proplatelets.
And once released, they circulate for a very short time, just 7 to 10 days.
They're constantly monitored for aging. As they get older, they expose specific galactose residues on their surface, which are recognized and phagocytosed by specialized macrophages in the liver called Kupffer cells.
The platelet's internal organization, visible by TEM in figure 10.17, is highly structured into 4 functional zones.
The organization is key to their rapid response. First, the peripheral zone. The cell membrane and its thick glycocalyx, which has receptors and adsorbed clotting factors. Second, the structural zone, the internal cytoskeleton, defined by the marginal band of microtubules that maintains that crucial discoid shape, along with actin and myosin for contraction.
The third zone, the organelle zone, holds their ammunition, the 3 types of granules.
This is where the power is stored. The alpha granules are the most numerous. They contain components for long-term repair, including fibrinogen, coagulation factors, and powerful growth factors like PDGF, platelet-derived growth factor.
The next ones, the dense or gamma granules, are the immediate responders.
They are smaller, containing non-protein factors needed for activation and recruitment. ADP, ATP, bioactive amines like serotonin, a potent vasoconstrictor, and, crucially, calcium ions.
And the lambda granules handle the cleanup.
They're lysosomes, containing hydrolytic enzymes used later for dissolving the final clot. Finally, the fourth zone is the membrane zone, which houses the open canalicular system and the dense tubular system, which is the storage site for that crucial intracellular calcium.
All this complex structure is geared toward their ultimate purpose, hemostasis, or controlling bleeding.
Hemostasis is a highly choreographed sequence. When the endothelium is injured, the underlying connective tissue gets exposed. This exposure triggers rapid platelet adhesion and immediate degranulation. The release of serotonin from the delta granules causes a rapid, temporary vasoconstriction to reduce blood flow.
And then the recruitment process begins to form the first physical seal.
The released ADP and thromboxane A2 are potent aggregators. They recruit more platelets to the site, leading to the formation of the unstable, temporary primary hemostatic plug.
And at the same time, the coagulation cascade gets a boost.
Activated platelets provide a crucial catalytic surface, which accelerates the local coagulation cascade.
This cascade culminates in the formation of the definitive seal, the secondary plug.
That platelet surface activity facilitates the massive conversion of fibrinogen to insoluble fibrin. And this fibrin rapidly forms a dense cross-linked mesh, shown in figure 10.18, that traps more cells and platelets, creating the stable, definitive clot.
And when the vessel is repaired, the clot has to be dissolved.
Clot retraction happens via the action of platelet actin and myosin. Clot lysis, or dissolution, is mediated by the enzyme plasmin, which is activated from its inactive form, plasminogen, by tissue plasminogen activator, or TPA.
And TPA is now a standard clinical tool, isn't it?
It is. Synthetic TPA is administered to break down inappropriate blood clots, most commonly in the treatment of acute ischemic stroke. And furthermore, those growth factors like PDGF released from the alpha granules play a crucial later role in stimulating the surrounding cells to divide and support long-term tissue repair.
We've now detailed every single formed element. To assess this complex system clinically, physicians rely on the complete blood count, or CBC.
The CBC is the universal diagnostic panel. Today, it's usually performed by automated flow cytometry and provides rapid quantitative and qualitative data on all the circulating elements.
Let's quickly review what those numbers indicate, starting with the total leukocyte count, the WBC.
An elevated WBC, or leukocytosis, is a general sign that the body is mobilizing its defenses: inflammation, infection, severe stress. At extreme elevation, hyperleukocytosis is often highly suggestive of leukemia.
And a low count.
A decreased count, leukopenia, is a grave sign, often linked to bone marrow failure, chemotherapy, or severe autoimmune conditions.
And the WBC differential provides the essential context.
The differential gives you the percentage breakdown of neutrophils, eosinophils, basophils, lymphocytes, and monocytes. Critically, it also includes the percentage of immature forms like band cells. An increase in band cells, often called a left shift, is a classic sign of an acute bacterial infection.
For the red cell line, we look at the RBC count, HCT, and HGB concentration.
An increase in RBC count is polycythemia. HCT and HGB remain the two most reliable indicators for assessing the severity of both polycythemia and anemia.
The erythrocyte indices then refine the diagnosis of anemia.
Correct. They give us the specifics. MCV tells us the average size of the cell. MCH tells us the average hemoglobin content per cell. And MCHC tells us the average hemoglobin concentration within the cell. The RDW measures the variation in size and shape.
And finally, the thrombocyte or platelet count.
High platelets, thrombocythemia, can be due to inflammation or proliferative disorders. Low platelets, thrombocytopenia, risks spontaneous bleeding.
All these formed elements have finite lives, meaning they must be continually produced, the definition of hemopoiesis.
Hemopoiesis is the essential balancing act of continuous cell production and destruction, covering erythropoiesis, leukopoiesis, and thrombopoiesis. It's a massive, tightly regulated system.
Tracing the historical development of this production is fascinating, starting in gestation.
Figure 10.21 illustrates the three sequential phases. It begins very early in the third week with the yolk sac phase. This quickly transitions to the hepatic phase, where the liver is the major site during the second trimester.
And the final shift moves production to the bony structures.
The bone marrow phase begins in the third trimester. And post-birth, the bone marrow becomes the exclusive site of blood cell formation, aside from lymphoid tissues.
The guiding principle for this complex system is the monophyletic theory.
This theory dictates that every single blood cell, regardless of its lineage, originates from a single common ancestor, the Hemopoietic Stem Cell, or HSC. This HSC has two key abilities: self-renewal and multi-lineage differentiation.
The HSC quickly commits to one of two major lines of progenitors in the bone marrow.
The commitment splits into the Common Myeloid Progenitor, CMP, and the Common Lymphoid Progenitor, CLP.
The CMP leads to the majority of circulating cells.
The CMP ultimately gives rise to megakaryocytes, erythrocytes, monocytes, and all three granulocyte types. The CLP is simpler, focusing on B cells, T cells, and NK cells.
And this entire massive process is governed by chemical instructions, the cytokines, and growth factors.
Absolutely. Cell proliferation and maturation are tightly controlled by these specific signaling molecules, many of which fall under the umbrella of colony-stimulating factors, or CSFs.
Let's dedicate some serious attention to the detailed development stages, starting with erythropoiesis, shown in figure 10.22 and plate 10.3. This is a dramatic loss of organelles in the nucleus.
The entire process takes about a week. We begin with the proerythroblast, the largest precursor, with a big nucleus and highly basophilic cytoplasm due to massive ribosome production.
It matures quickly into the basophilic erythroblast.
The stage is smaller, the nucleus starts to condense, and the cytoplasm is intensely, almost violently, basophilic because the cell is maximally producing polyribosomes to synthesize hemoglobin.
Then comes the stage where the color literally changes.
The polychromatophilic erythroblast. The cytoplasm shows both basophilia from the ribosomes and eosinophilia from the hemoglobin, giving it a mixed gray or lilac color. The nucleus shows a characteristic checkerboard chromatin clumping, and this is the last stage capable of mitosis. Cell division stops here.
After this, the cell prepares for life in the circulation by ejecting its entire command center.
That's the orthochromatophilic erythroblast, or normoblast. The cytoplasm is now fully eosinophilic because it's packed with hemoglobin. The nucleus is tiny, dense, and extremely condensed. Late in this stage, the nucleus is extruded from the cell.
And the final stage released into the blood is the reticulocyte.
The nucleus of cell that enters the circulation is the reticulocyte. It still has some remnant polyribosomes and mitochondria. It matures in the blood for about 24 hours. A normal reticulocyte count, about 1 to 2% of total RBCs, is a strong indicator of healthy marrow activity.
So we can summarize this entire week-long process by observing 5 visible trends, which you can see in figure 10.23.
If you look at the progression, you see 1. Decreasing cell size. 2. Decreasing nuclear size with increasing chromatin condensation. 3. The disappearance of nucleoli. 4. A radical shift in cytoplasmic color from dark blue to pure pink. And 5. The entire nucleus gets ejected.
Regarding thrombopoiesis, we just need to reiterate that it's driven by TPO, moving the CMP through to the massive megakaryocyte, which then fragments into platelets.
And its clinical importance is profound. Thrombocytopenia low platelets is a risk factor for spontaneous bleeding.
Let's detail the maturation of the white cells. Granulopoiesis, shown in figure 10.22 and plate 10.4. This takes about 2 weeks. The commitment to a specific lineage doesn't happen until the myelocyte stage.
The starting point is the myeloblast, the first recognizable precursor. It has a large euchromatic nucleus and basophilic cytoplasm. It rapidly matures into the promyelocyte.
The promyelocyte is a landmark because it is the only cell in the lineage that synthesizes those large, dark, azurophilic primary granules.
That is the crucial distinguishing characteristic. Since the cells continue to divide after this stage, the number of primary granules gets diluted through subsequent divisions. This leads us to the myelocyte.
The myelocyte is the point of lineage commitment.
It's the first stage where the unique lineage-specific secondary granules become visible. The nucleus begins to indent, and the color of these granules determines the lineage. Light pink for a neutrophil, large red ones for an eosinophil.
Next, the metamyelocyte.
The nucleus deepens into a distinct kidney bean or horseshoe shape. This is followed by the band stab cell, where the nucleus is uniformly horseshoe or elongated. An increased percentage of Gann cells in the peripheral blood is that left shift we mentioned, signaling an acute bacterial infection.
The kinetics of granulopoiesis are fascinating because the bone marrow maintains this massive reserve ready for instant mobilization.
The marrow stores a pool of mature neutrophils that is 5 to 30 times larger than the entire circulating population. This is maintained by an exquisite regulatory mechanism centered on the CXCL12-CXCR4 axis.
How does this molecular lock-and-key system work to keep that army in the barracks?
Stromal cells in the bone marrow produce the chemokine CXCL12. Mature neutrophils express the receptor, CXCR4. When CXCL12 binds to CXCR4, it generates a strong retention signal that arrests the mature neutrophil in the marrow space.
So when a bacterial invasion triggers inflammation, that signal must be released.
Acute inflammation releases cytokines like GCSF. These signals suppress the production of CXCL12, removing the retention signal. This instantly triggers the rapid release of that massive reserve pool into the circulation.
And the same receptor seems to be used as a homing signal for the neutrophil's final cleanup.
Yes, it's a master switch. As neutrophils age, they upregulate CXCR4 expression, which facilitates their migration back to the marrow, liver, or spleen, where they undergo apoptosis and are destroyed.
This molecular axis is perfectly illustrated by the clinical example of WIMM syndrome.
WIMM syndrome is caused by a genetic mutation that prevents the CXCR4 receptor from being internalized after it binds CXCL12. This prolonged, constant signaling enhances the retention of mature neutrophils in the marrow.
So they're produced, but they can't get out.
Exactly. The result is, despite a healthy production and a massive reserve, the patient suffers from chronic neutropenia because the neutrophils are perpetually locked inside the bone marrow.
Finally, we must briefly mention the development of monocytes and lymphocytes.
Monocytes derive from the GMPs, influenced by MCSF and GMCSF. Lymphocytes from the CLP line mature according to their type. T cells in the thymus guided by γ3, and B cells in the bone marrow or lymphatic tissues guided by PAX5.
And we can't overstate the importance of the regulating glycoproteins detailed in Table 10.4.
These CSFs and interleukins are produced by a vast array of cells and target specific progenitor stages to precisely control the entire rate of hemopoiesis. Understanding this cascade is why recombinant growth factors, like GCSF, are essential after chemotherapy to boost a patient's white cell production and prevent lethal infection.
Our final segment focuses on the physical location of this spectacular process, the bone marrow itself. We start by distinguishing the two types of marrow.
In adults, we distinguish between red bone marrow, which is the active site of hemopoiesis, found primarily in the flat bones, skull, and vertebrae. Then there is yellow bone marrow, which dominates the medullary cavities of long bones.
And that's mostly fat.
It's inactive, composed mostly of adipose cells, though it retains the potential to revert to active red marrow during periods of extreme demand, like massive blood loss.
Looking at the microscopic architecture in Figure 10.26, the marrow isn't a solid mass, it's organized around a specific vascular structure.
The functional architecture consists of hemopoietic cords, the areas of intense cell proliferation, separated by specialized thin-walled vessels called sinusoids.
And the wall of that sinusoid is unique.
It is. It has an endothelial lining, a discontinuous basement membrane, and an incomplete external covering of adventitial reticular cells. These adventitial cells are crucial. They provide physical support and are the main source of the regulatory cytokines that stimulate the progenitor cells.
The entry mechanism into the circulation is highly regulated and uses a closed circulation system.
This is a critical barrier. Mature blood cells can't just squeeze between the endothelial cells. Instead, they must use the transcellular diapedesis pathway, meaning they have to actively pierce through the cytoplasm of the endothelial cell itself.
And within those hemopoietic cores, the different cell lineages develop in very specific neighborhoods.
Development is highly spatial. Erythrocyte nests, or erythroblastic islets, are often found clustered immediately adjacent to the sinusoid wall, frequently surrounding a central macrophage that helps process the extruded nuclei.
And megakaryocytes are right there too.
Right next to the wall for easy platelet release. Granulocytes, conversely, develop farther away in the center of the cord and migrate to the sinusoid only when they're fully mature.
This structure leads us to clinical correlation 10.6, assessing bone marrow cellularity. This is a key diagnostic measurement.
Cellularity is defined as the ratio of active henopoietic cells to inactive adipose cells, and it's age-dependent. Our sources provide a useful formula: 100 minus the person's age, plus or minus 10%. So a healthy 70-year-old should have a cellularity between 20 and 40%.
So if a 70-year-old has 85% cellularity, that's dangerously high.
That's hypercellular marrow, often indicative of a proliferative disease like acute myelogenous leukemia. Conversely, very low cellularity, like 5% in an adult, is hypocellular marrow, typical of aplastic anemia where production is failing entirely.
And finally, the two techniques used for examining the marrow.
Figure 10.27 shows the two methods, which are usually performed together. The aspiration uses a needle to extract liquid marrow, which is then smeared. This is vital for analyzing the fine morphology of individual cell types, crucial for leukemia diagnosis.
And the core biopsy.
The core biopsy, or trephine, extracts an intact piece of bone and marrow, which is fixed and stained with H&E. This is essential for assessing the overall tissue architecture, spatial relationships, and, critically, the absolute cellularity.
This has been an incredibly detailed look at the histology of blood. Let's quickly summarize the critical takeaways from this deep dive.
Blood is a specialized fluid connective tissue made of plasma and three primary formed elements. Erythrocytes are masters of gas transport, relying on their biconcave shape and that complex spectrin-based cytoskeleton.
Leukocytes are the core of immune defense, categorized by their granules, with neutrophils using those explosive chemical strategies like the MPO-driven respiratory burst.
And platelets, fragments of megakaryocytes, orchestrate the entire hemostasis and vessel repair process using their internal granules.
And every single one of these cell types is continually and meticulously renewed through hemopoiesis in the red bone marrow, a process governed by specific progenitor cells, transcription factors, and a precise cascade of cytokines.
Right. And as a final thought, step back and consider that neutrophil homeostasis system again, that CXCL12 and CXCR4 interaction. Think of the elegance of that single molecular retention signal.
It holds the army in reserve.
It maintains a ready-to-deploy reserve that's 30 times the size of the active patrol, demonstrating this exquisite balance between massive supply and instantaneous demand, all managed by one protein complex. That level of control, operating unseen at the microscopic level, is truly the marvel of cellular biology.
That remarkable efficiency is a perfect illustration of the elegance beneath the surface. Thank you for diving deep with us today.